Here's an overview of the lecture we're about to engage in. We're going to talk about protein function, and I'm just going to introduce a couple of ideas that govern protein interactions with ligands and with small molecule ligands, for example, and with each other as well. Complementarity is going to be key, as the Pauling article that I gave you to read indicates, and we'll also look at how prosthetic groups affect protein function or extend protein functionality, and we'll look at some models for interactions between proteins and ligands. We're going to use two molecules in these lectures as archetypes of protein function. Myoglobin is a simple model because it doesn't involve any real conformational changes when it interacts with its target oxygen. Hemoglobin, on the other hand, is a much more complex system, but because of its similarity to myoglobin, it makes a good extension of the simple to the complex. So, there we go. Okay, so function derives from structure. Complementarity of interactions determines how well two molecules interact. This is the basis of Pauling's hypothesis that you've all read about by now. Complementary surfaces govern protein-ligand interactions. So how well the three-dimensional topology of the structure matches, one structure matches another, how well electrostatics match one another. Binding site chemistry and sterics then of the protein must match that of the ligand. That's the idea of complementarity. However, we don't think of complementarity in a rigid sense. Proteins and ligands both undergo conformational change, and so one can imagine that the protein could induce a conformational change in its ligand, or you could also think of that as the protein binding site stabilizing a particular conformation in the ligand, and the same is true of the ligand altering the conformational state of the protein. We've talked about this already in terms of how, for example, some drugs can be used to stabilize proteins against degradation or amyloid formation. So in that case, the drug is changing the conformational state of the protein. Another way to think about that too, though, is to say that there might be conformational states that are in equilibrium in the absence of the ligand, but the ligand itself changes that equilibrium or shifts that equilibrium towards one particular state, that is the bound state. Easiest way to think about it, though, is simply through lock and key interactions. And the only reason I'm showing you this DNA binding protein and its ligand DNA is that we see how intimate those lock and key interactions can be, realizing, though, that this is just one extreme, and that induced fit is the other extreme. So perfectly complementary surfaces encounter each other and lock together. So these are highly specific interactions, and in this case I'm just showing you a viral fragment interacting with a fragment of an immunoglobulin, and this would be the hypervariable region of that antibody, for example. These interactions are stabilized by hydrogen bonding, hydrophobic effects, salt bridges, so electrostatic interactions of many flavors. Again, only showing you here how intimate those interactions really are. Oh yeah, so if you want to orient yourself, we're just looking at that hypervariable region of the antibody interacting with a piece of the viral coat, I believe. Highly specific interactions. However, those specific interactions those interactions can be for conformations that might not be populated in the absence of the ligand. That is to say, again, that we might have multiple states that we can populate, and the bound state we almost never populate in the absence of ligand, but only when the ligand is present do we shift the equilibrium toward that state. is one way to think about it. And in this case, we're looking at hexokinase, which is an enzyme involved in carbohydrate metabolism. You might guess from the name that it phosphorylates a hexose sugar. So kinase indicating that it phosphorylates something. Hexo indicating that the ligand is a six carbon sugar. In this case, what I've done is superimpose two hexokinase structures. Both of these structures are from the Stites lab. We see in red the change in one domain with respect to another. So there's a sort of a hinge that is flexible and we select for one conformation. So here's in blue we have the open conformation. And when there's no ligand present, and when the ligand is present, then we move. And this might look like a small change to you, but it's not what in terms of what the carbohydrate sees. We move when we have that carbohydrate present. So new chemical information in the way of hexose. Right, there's the sugar equals new structure. Now, this is pretty interesting as well because it allows us to select what kind of substrate we might bind and interact with. Right, so plenty of hexoses might fit in the active site, but relatively few are going to induce the conformational change, or at least induce it to the extent that the natural substrate does. So in that sense, it's a regulatory strategy. We don't phosphorylate just any old carbohydrate. We only phosphorylate the ones that satisfy the proper complementary interactions. Here again is just another cartoon of that induced fit sort of interaction. On the left, we see an antibody with no antigen bound, and what's given to you as reference points are these two blue and one red structure. It doesn't matter what they are. You can clearly see that that's probably a tyrosine. I hope you can see that. And when we have the ligand present, which is probably some peptide analog, doesn't really matter. We see that the relative positions of those change. Not only, and so we see two things really. In the free state, we see this deep crevice, and if we were staring at that, we might actually think, well, that looks like it might be a good binding site for something. And then when we actually place something in that crevice, we see that the structure does alter, and the relative positions of all of those molecules change. So, for example, if we look closely, we see that these two structures have shifted dramatically apart from one another to perhaps accommodate the binding of that substrate. So, induced fit. I should say that it's most likely that any interaction is going to involve some kind of induced fit, some kind of change in the structure of one or both partners. And a rigid lock and key sort of model is a nice conceptualization, but rarely encountered. So let's look then at our first example, myoglobin. This is the globin. We've seen the globins before, right? It's a sort of a complicated helical bundle that serves to anchor a heme prosthetic group, which itself binds an iron 2 or ferrous iron, and there's an oxygen molecule bound at the iron. Yes, and there's our nomenclature, our labeling scheme. A couple of you are asking me about this. What's this F helix? What are these helices that are involved? Well, it's just simply N, right? So here's the N terminus, A, B, C, D. It's just N to C terminal labeling of the helices as they occur in the sequence. Not really that terribly important, but we will sometimes see different molecules or different histidines referred to by their position with respect to those helices. So anyway, myoglobin's job, well, it's an oxygen storage molecule. It exists at high concentration in muscle tissue and serves to maintain oxygen concentration for respiration. Iron is bound at the heme. So let's just spend a minute talking about those prosthetic groups like I promised you. The hemoglobin and myoglobin both involve a porphyrin, happens to be iron protoporphyrin 9. It doesn't matter, but I'm just giving you a sense of that there might be many variations on this structure. But in general, a porphyrin is a fully conjugated tetraperol ring. So here's a perol, right? Those of you remember that from your organic chemistry, linked with these methylenes, right? or methynes, yeah, and we have a fully conjugated system here. So what do you think that means? Well, we have a large conjugated pi electron system. That's going to mean strong absorbance, and of course we know that hemoglobin and myoglobin are reddish or purplish depending on their what ligand they've bound. But in general, these porphyrins strongly absorb in the visible range. We can add a ferrous iron atom bump right there, and converting the free base here to an actual heme. So this is a porphyrin. Now we have a heme, and we can now the heme is free to bind its ligand oxygen. So here's iron protoporphyrin 9, and you can see that we've decorated the porphyrin with various molecules or various functional groups. There are a variety of other prosthetic groups, and we don't have time to go over them today. You will talk more about them in the second semester of biochemistry if we see you there. But let's just take a couple of examples. So prosthetic groups, of course, as we know, enhance the chemical and binding properties of proteins. The heme of hemoglobin and myoglobin allow it to position an iron atom such that the iron atom can interact with oxygen. There are other ways that you could do that, though. Hemocyanin, an invertebrate oxygen carrier, has two copper ions bound to that are immobilized with a series of histidines. So, and in this case, the copper center is actually the oxygen carrier. And let's see. Oh, chlorophylls, right? So this is a little bit difficult to see. I apologize for that. But again, you can see this is another porphyrin, right? And now it's highly modified, right? We've got an extra ring here. Our pi system is perturbed. We have a magnesium at the center instead of an iron atom. And what does this do? Well, it dramatically changes the absorption maximum. We all know what the absorption spectrum of chlorophyll looks like. The stuff is green, and that arises from these complex and relatively subtle changes in the structure of the porphyrin itself.